In this video, we will continue our discussion about how this VCAM-1 mediates VCAM-1 mediates the proliferation and the cell cycle checkpoint at G1-S in the pancreatic cancer cells. In the previous video, we have discussed that VCAM-1 has been overexpressed in the pancreatic cancer patients. So, based on the results of the previous finding, they further decided to focus on this VCAM related changes in the pancreatic cancer cells. So in the panel A, they did they designed a silencing RNA that can suppress the VCAM overexpression, or that can suppress the VCAM expressions in the pancreatic cancer cell lines. That can be seen in the panel A and the panel B, where they can see that in Pancreatic and the KPN2, the VCAM expressions are being reduced compared to the control silencing RNA. And same things they have seen in the case of Western blot, where the expression of this VCAM has been reduced when they use silencing RNAs. And this panel A is about the suppression of this VCAM1. However, they also developed a system where they can overexpress this VCAM1 using this vector, that is pcDNA 3.1-VCAM1. This helps in overexpression of this VCAM in the PANC1 and also in the KPN2, and these things are validated using Western blot also, that can be seen here. And after developing this system, they wanted to check the proliferations in the PANC1 and the KPN2 cells in both the systems. And what they have found out is that when they use the silencing RNA, there is a suppression in the there is a relative decrease in the proliferations compared to the control case for the both PANC1 and KPN2. And when they use it for this VCAM overexpression system to check the relative proliferation, they see that proliferation increase with the overexpression of this VCAM both for the PANC1 and the KPN2. And all this indicates that VCAM is directly helps in the proliferation of the pancreatic cancer cells. And they have validated their results using the colony formations also, where they can see that when they use the silencing RNAs, the colonies formations are low compared to the control silencing RNAs. And when they use the silencing RNAs for the VCAM1 in both the PANC1 and KPN2 shows low colony growth compared to that control silencing RNAs, which does not silence the VCAM1. And similarly, with the overexpression of this pcDNA 3.1 VCAM1, they see that more colonies in the PANC1 and the KPN2 compared to the other control cases. And these things are quantified on the right hand side, where we can see that the decrease is statistically significant when they use silencing RNAs, and the overexpression is also statistically significant when they use this pcDNA 3.1-VCAM1 plasmid. And after validating their results using colony formations, they wanted to check whether what effect it has on the cell cycle. And when they checked the cell cycle assay using propinium iodide, what they have find out is that there is a decrease in the S phase when they use silencing RNAs for both PANC1 and KPN2. The quantified is on the in the panel G, where we can see that decrease in the S phase in the both PANC1 and KPN2 cell lines, and simultaneously increase in the G0 G1 phase for both the cell lines. And when they checked it for the overexpression of this PANC1 using pcDNA 3.1, they have find out is that there is an overexpression of the S phase in both pcDNA in both PANC1 cell lines and KPN2 cell lines, where they use pcDNA 3.1 pcDNA 3.1 VCAM1 plasmid, indicating that with the proliferations there is an increase in the S phase, and with the decrease in proliferations or decrease in VCAM1, there is a increase in the G0 and G1 phase. So all these results indicates that VCAM1 helps in the proliferation. At the same time, it also affects the cell cycle at G1-S checkpoint. So I hope you understood what in brief about the results that are presented in this figure. And if you need to understand these experiments in detail and their analysis in more detail, you can go through the reference that has been provided in the description, and where you also have the access to the supplemental figures. So that's all for this video. Thank you for your time.